So there could be some other features that are present in plant, like you can say that no doubt that plants are classified on the basis of this. But what about the rest of the classification, like subgroups of plants? How are we going to classify the subgroups of plants? This is one of the very important statement. You can refer this as statement, or you may refer it as the concern. But yes, the plants have to be classified further because there is a very wide variety in plants that we can find. So obviously plants, the name refers to basically the kingdom. You can simply say that plants are the kingdom. The name plants is the name of a kingdom. Then after that, further, um, the what about phylum, class, like the groups that we have been studying in the animals? Are we going to study those groups in plants? Yes, we are. We are going to study those groups in plants. And how? Let's see what we can do here. Number one, the phylum that we often um, you can say study under the plant. There are many phyla. Phyla means plural. There are many phyla in the plants. Or you can say there is a vast variety of phyla in the plants. But we mostly refer only to a phylum called as Tracheophyta. Now, this word may be missing from this particular book, but this group basically has to be explained to tell you what kind of organisms we are going to study. See, Tracheophyta is a kind of plants that contain xylem and phloem. Now, this would be a bit, you can say, a bit surprising to you. This would be a bit surprising to you, but remember that there are plants who lack the xylem and phloem. There are plants. So that is why we say that Tracheophyta is the phylum that contains the xylem and phloem. Let me mention you as an extra information the group that does not contain the xylem and phloem. This group is called as Bryophyta. Just to tell you the comparison, you don't need to remember it. Okay. So next is um, after the Tracheophyta, the classes. Now, about the classes, you need to remember that we are going to talk about three different classes. One class of the plants is called as ferns, and the other class of the plant is called as angiosperms, or hold on. Ferns are those plants which are seedless. They don't make the seed. But there are other plants which are seed producing plants, and these are called as spermatophyta. Sperm means the seed, the phyte or phyta means plant. Spermatophyta. That is a group or a class of the plant that makes the seed. Then there are subclasses. Subclasses of the plants. After spermatophyta, there are subclasses of the plant that we often refer to as the angiosperm. You may have heard this name before, and the gymnosperm. Okay. The angiosperms are those plants in which seed is within the fruit, within the fruits, while the gymnosperms are called as conifers. Basically, seeds are out of fruits, are without fruit. Sorry, not sorry, are not out of fruit, without fruit, without fruit. You may have seen. Have you ever been to Pakistan?
Yeah.
Any hilly station?
Hilly station.
Any hill station? Yeah,
Um, I don't really know what you mean.
like mountains,
Yeah, yeah,
like
yeah.
Murray, or
Margala.
you may have. Yeah, Margalas. Now you may have seen some particular plants having the cones on them. Those cones are basically belonging to gymnosperm. These plants never form the fruit. They would just make a cone, a woody textured cone on which the seed would be present. So, like the gymnosperms are not our real concern. Talking about the class, the spermatophyta, you can just remember that this is a subclass. Angiosperm is a subclass and gymnosperm is a subclass. Talking about angiosperm, in angiosperms, we have two families. One is called as monocotyledons, and the other one is called as dicotyledons. These are two families. Now, I'm just making this whole, you can say, flowchart to make you aware that in what way we are classifying these things, and what do we mean by all this stuff, and what are different purposes of these names, and what are different meanings of these names. Got it?
Yeah.
Just look at the classification that I just drew here, then we'll continue.
Yeah. Okay.
Okay, moving ahead, we are going to talk about different kinds of features. Okay, so the plant kingdom, okay, the main groups are algae, mosses, ferns. Basically, algae are not in plant kingdom. Remember, these are not in plant kingdoms. Just to make the things easier, they have written it over here. Yes, mosses. These mosses are the same ones I told you about, the bryophytes. They don't have xylem and phloem. And ferns and seed plants, they have xylem and phloem. Okay, now this is a fern plant. This is a fern plant. And a fern plant is having certain features on it. So let's talk about those features. They are much better adapted. They have roots, stems, complex leaves, and vascular tissues. And like obviously, the the group before the ferns were mosses. They were not containing complex leaves. They were not containing xylem, phloem. They were not containing proper roots, stems, and leaves, etc. So, so basically, they are much better adapted. Then then they are able to produce spores for wide dispersal. Okay, what do you know about the spores?
Um, spores. So like they're not able to s- produce the spores for wide dispersal. Basically means that
They are
they
able to.
can't.
They are able to.
They are able to produce spores for wide dispersal.
Hmm.
So that so this is their way of reproduction basically. That they drop the
Like,
spores to
hmm
grow
hmm
trees right next to it or further away.
hmm okay, spore. A spore basically is a very lightweight. You can say seed-like structure. I would never say it's a seed, but it's a seed-like structure, and it can be dispersed through wind. Dispersed through wind. Whenever it disperses through wind, it can grow far away. It can grow closer or far away. The what is the requirement? The requirement is the ideal condition. If the conditions are ideal, they would grow closer, and if the conditions are not ideal, they would grow much far. They would not even grow. They would not even grow. So uh, that is basically the theme of it. And spore is, however, they do not have very thick cuticles and can only survive in shady and humid areas. Now, this is very important feature. Remember that the the cuticles, what are the cuticles? Cuticles are wax layers, waxy layers. You may know already that
Yeah.
they are wax layers. Now, the wax protects the evaporation or the loss of water. They would be like whenever you would put a fern. We are talking about fern right now. When you would put the fern under the sun, they would immediately die. Reason is because the ferns are actually very delicate. Their water evaporates immediately. Okay. Uh, Another thing, the gametes of ferns, like those of mosses, must swim through film of moisture to reach the site of fertilization. Like they cannot reproduce cannot reproduce without water. There was another group in animals which could not, which although used to live on the dry land, but it cannot reproduce without water. What was that group?
Um, reproduce without water.
Without water.
That can or cannot.
They cannot reproduce.
Um, cannot reproduce. Was it bryophytes?
No, 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 no. Yeah, in plants they are bryophytes, but what about the animals?
Um, was it like parthenogenesis, something like that you
No,
mentioned?
no, no, no. These organisms are called as amphibians. These organisms are called as amphibians who can survive on land, but they cannot reproduce without water, because their gametes have to be transported through water from male to female. That is basically the idea of reproduction inside water, or they cannot reproduce without water because the sperm has to swim. It has to swim towards the egg. That is the reason. Okay, so one of the examples here is the one of the examples here is this organism, this particular plant, and this the name of this plant is although not written here, it is called as adiantum. So this plant, first of all, remember their stems are underground. Understand this one. There are many plants in around us that have stems underground. Many plants. Like any plant, do you remember that has stem that is underground?
Mhm.
One of the most famous plant around us that has stem underground are, have you ever seen the stem of grasses?
Yes.
When?
Like when you pull out the grass, then you see the roots, right?
Yes. You can never see the stem of the grass until you uproot it, until you remove it from the ground. You will see brownish stem, and covered under the soil. That is basically the the stem of the grasses. So, like that, they also have the stem underground, and below the side of the stem that is towards the soil would grow the roots. It would grow the roots. What is the meaning of true root? True basically refers, it is a comparison between the bryophytes and these plants, between bryophytes and ferns. Bryophytes don't have true roots. They don't have true roots. They just have extensions like roots, but they don't have true roots. Then after that, this is a leaf. This whole thing is a leaf, and this actually uh, particular leaf is not a leaf. It is called as a frond. Remember that it is not called as a leaf, although we can see it as a leaf. We can name it commonly as a leaf, but it is not a leaf. This particular whole thing that has come underground, it is called as a frond. And uh, these are each of these are leaflets. You cannot call them leaves. These are leaflets. And the these are now why it is calling it as a stem. This is not a stem, by the way. This is just a branch containing the xylem and phloem. Remember that mentioning the rhizome and stem separately, it's a mistake. Rhizome is a stem. That's all. There is no other stem. So remember that the stem is only this one, the underground one. This is just a branch. And you see this one looking like a snail, looking like a snail. What is this? See, remember that this is called as a young frond. And you know what happens when they would be they would be turning into this whole large frond. These small fronds, they would be turning into large fronds. How? A small frond look like this. It would be rolling up, rolling itself, rolling up itself. So that is what a frond look like. Now, the most important thing that you would see in this particular plant, this particular plant is having these leaflets, and on the underside of these leaflets, you have orange, orange circles, and these orange circles are basically the sporangia. Sporangia, and remember that sporangium or sporangia, they are special bodies, special bodies holding spores. They hold the spores. So remember that the sporangia are present under the leaf and the spores are present inside the sporangia. And when they burst, the spore just move out. They burst, the spores just move out. That is why they are called as sporangia. And they are showing you this that thing over here. Okay, moving ahead, entering into the realm of the angiosperms. Okay, so more than 80% of all plants are angiosperms, and the diagram at the top of the opposite page. Okay, two groups are monocotyledons and dicotyledons. Now, this is important. What's a cotyledon?
A cotyledon.
Yes.
So is that the part of the leaf that has the plant inside the plant that the seed is growing, or it's like
No.
producing stores food?
Okay. Hold on. Hold on. Just give me a minute. So, let me tell you about the cotyledon first. The cotyledon, it's basically a leaf of a seed. Remember that obviously when the plant is mature, it makes the seed. Sorry, mature plant has leaves, right? Mature plant has leaves. But remember that when a plant is a seed, suppose that this this is a seed, this is a seed, and this seed would be containing the leaves inside. These are, you can say, these are the first leaf that would be performing the photosynthesis when the seed would burst and go above the soil. Right now it is underground. It is underground, but still the leaf has the seeds has have leaves. Suppose, let's talk about human embryo or human baby. Does it have hands before birth?
The hands develop.
Yeah,
So
it
when it's
develops.
born, it has hands.
Yes. The last organs that mature or develop in a human baby are the lungs. But still, before birth, the lungs are there. The lungs basically develop themselves when the baby comes out and they start interacting the air. Because inside the baby, there is no way the baby can interact with the air. But still, it does not mean that the baby would born and the lungs would start sprouting after the birth. No, we make the lungs. Our lungs are made before birth. Our lungs are made before birth, and then then you can say they are made before birth, and then we use it when we are born. Similarly, the seeds always have the leaves in them. Before sprouting, before coming out of the soil, they have leaves in them. Now, some seeds have two leaves, and some possess one leaf. Those having only one leaf would be called as monocotyledons, and those who are containing two leaves would be called as dicotyledons. Got it?
Yeah.
But there are other features. There are other features uh, that seen can be seen in a monocot or dicot. In a monocot or monocotyledon or dicotyledon, what other features you can see? Have a look. In monocotyledon, you have one cotyledon, and okay, they would have parallel veins. Parallel veins mean that it is a leaf and its veins would never cross. Veins would never cross. These are called as parallel. When you would when you would see a dicotyledon leaf, they would have branched veins. Their veins would cross each other. Got it?
Yeah.
Another very important feature that you would find in the particular monocotyledons and dicotyledons is that in a dicotyledon, whenever a flower is made, each group of petals is a group of five or multiples of five, meaning 5, 10, 15, 20. This is for dicotyledon. In monocotyledons, a, uh, a flower, when it is made, it either would be a multiple of 3, 3, 6, 9, 12, or a multiple of 4, or a multiple of 4, 4, 8, 12, 16. That is for the monocotyledons. For dicotyledons, you will find the 5 or multiple of 5 flowers. That is a very important feature that they are missing in this particular chapter. Got that?
Yeah.
This basically is an angiosperm. An angiosperm, like they are telling you the common structure of all angiosperm. Just have a look and read the labels.
Okay.
Just hold on a second. Okay. So now this was all the account we needed to study about the plants. Now we are going to talk about the other organisms. Like we have already been talking about the prokaryotic cell, what are bacteria, etc. We need to talk about the fifth kingdom. The fifth kingdom, I guess we haven't touched. No, it's not here. Yes, it should be. Have you studied about the prototists or no?
No, I haven't.
Okay, they have not mentioned the prototists over here. Let's leave it. Moving ahead. Now, um, how did I define the prototists, by the way?
Sorry, say that again.
How did we define the prototists before?
Oh, in when we were talking about the prokaryotes and
No, protoctist, not
yeah,
prokaryote.
so when we were
Protoctist.
talking about the kingdoms and everything.
Hmm.
So wasn't it
And mhm,
unicellular or multicellular organisms or
mhm,
simple multicellular organisms like
mhm.
algae and mold, I think you said?
No, mold is fungus. Fungus is totally different.
I think you said slime molds or something.
Now, I said the protoctist is a
Yeah.
group of organisms.
Like
Uh,
algae is a protoctist, right?
yeah, and these organisms basically are, you can simply say they are made or they are grouped because we don't understand how to classify them. So what do? We just put them in a common group called as protoctist. That is what we said about them. So, but the last group, okay, there is a very important group called as viruses. What do you know about this group? This was not included in the five kingdoms, right?
No.
Why?
Um, why? Don't they're not considered living organisms.
And why is that?
They don't have cells.
Well then, living objects, they don't have cells. They don't have cells, and since they have no cellular machinery, the uh, they cannot show the features of a multicellular organism of or any cellular or any organism. They cannot show. So that is why we cannot say that they are we cannot say that they are the living objects. What is their total structure? Their total structure, and this group is basically one of the most notorious one. What is the meaning of notorious? Like famous in a negative way. This is a notorious group. Why? They are parasites. Parasites, they are pathogens, and most importantly, they would always be intracellular parasites. Intracellular means that you would never see them outside the cell. You would never see them outside the cell. You would just see that they are attacking their host, they are attacking their host or their organism um, when they are inside the cell. They always show the activity inside the cell. That is why we are never able to classify them with the living objects. That is the basically the main theme. Okay, so all right. The the complete things that they often possess are like, first of all, this protein coat. The protein coat is called as capsid. This protein coat is called as capsid, and this capsid basically is common. It's common between all viruses. Between all viruses. So it's a protein made coat, and you will never see. this type of code inside any other organism, and it is very much useful for taking entry into the host. One more thing about the viruses is that their genetic material. Basically, this is the genetic material that actually causes the disease. This actually causes the disease. And you can also say that why? Because the genetic material is used to make the poison, or you can say make new viruses that can disturb the other that can disturb the other cells. So genetic material is important. Then this whole structure, in this whole structure, one thing is there, that is spike. Remember that spike are you can say they are landing parts. Landing parts mean, as we have already said, that the viruses are always intracellular. They are always intracellular. Since they are always intracellular, it means that they need to enter into the cell. To enter into the cell, they need the landing parts. They need the parts that can be used for landing purposes. That can be used for the landing purposes. So that is, these are spikes. And most of the time, remember that spikes are not always, you can say the landing parts are not always spikes. There could be any other thing, but the main thing about it is that it has to enter into the cell, so it has to land on the cell. And there must be the parts on the virus cell that could be looking like a spike. So that is what we needed to know. Okay, next. Hold on, just a moment. I'm sorry for today. Basically, there's a worker here working at my home, so I have to attend him. Just give me 2 more minutes. Okay, sorry. So we are done with this chapter.
Okay.
So did you hear what I said about just now?
About what? What do you mean?
Uh, About I said that there are workers working at my
Yeah,
home, so
yeah,
I have to attend.
yeah, yeah. I heard that.
Okay. So now we have a few questions here, but we would be working on these questions later along with the, you can say, rest of the test. We'll be working on these questions as well. Let's talk about, like, we have done much talking about the cells. And did we talk about the microscope, by the way?
No, we didn't.
Okay. You want to, or it's fine because microscope is usually very easy.
Do you think we should cover it or?
It's not that much difficult. It would just take 10 minutes. So let's have
Okay.
a discussion. Basically, when we talk about a microscope is an instrument that is, what is scoping? Scoping is just like watching, watching something, and micro means very small, very small things. We are watching them. So, or focusing on them. Actually, what happens that the principle of microscope is to magnify. We magnify different things. We enlarge them. We enhance them, enhance their sizes, and we try looking at those things which we cannot see with our ordinary eye, cannot be seen with an ordinary eye. So that is often called as a microscope. And when we started progressing in the science, we were just available with the light microscope. The main thing in a microscope is source of illumination. Source of illumination. Okay, why the source of illumination is so important?
Um, why is it important? Source of okay, source of illumination. So to see um the, the object more closely. So is
Hmm.
the source of illumination, because when I use the microscope, there's like a light on
Hmm.
the uh you know where the lenses are? The light is there. I remember. Is that the source of illumination in this case?
Source of illumination, yes, is to light up the thing. Okay, let's have another small discussion. Why light is so important? Why do we need light? Hmm?
Wait, so how would I explain it? Because it's an obvious answer, but how would I put it into words that just to
Just,
see, just
yeah, okay. We need the light to do what?
see?
To see the things, right? The It means that we cannot see until the light reaches our eye. So that means whenever you would observing something requires observing something from inside requires the light to move through it. Requires the light to move through it. Now, what is so important about this statement? Remember that when we when we start the light, when we see the light, when the sun sends the light rays, there are three different resources through which the light can be seen. Number one, when you would be looking directly at the source of light. Number two, when the light strikes on an object and then comes to your eyes. And number three, when the light moves through something and reaches your eye. Is that right?
Yeah.
So we are using the third phenomena so that what do we do? We have a source of illumination at the base of the microscope, and the light rays emerge from it. Light rays emerge from it, and they basically pass through the specimen. The specimen is placed on the stage. This is your specimen. We adjust the light so that the light can pass through it and reach our eyes so we can observe it. That is why we name our this microscope as the light microscope. What is the other source of what is the other type of microscope?
I'm not sure.
There is another type of microscope that we often say at light microscope, electron microscope. Sorry, electron microscope, and you may have already known that electron microscopes are really important. Anyways, the important part is that these light microscope would be used, and remember, the whole initial discovery, the whole initial discovery was done through this microscope. This is very important to know. Whenever you would do, whenever you would see many things, many of the objects are named. Many of the objects are named on the basis of the observation under the light microscope. Let's suppose, let me tell you. Do you know about the mitosis and meiosis?
Mitosis.
Mitosis and meiosis.
Uh, I know what mitosis is. The mitosis is the when the cell replicates the chromosome.
Hmm. Yeah, enough. The microscope, it was the light microscope through which the mitosis was initially observed. It was not the electron microscope, and we named all the stages of it. There are many stages of it, but in junior classes, you may have only studied about one or two stages. It has many stages, and all of those were studied on the basis of the light microscope. So it was a very huge discovery. Right. There is an important part related to exams that let me tell you. Any picture, any picture taken from the microscope, that picture is always called as photomicrograph. 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 Photo means photo is photo, micrograph. Okay. So the photomicrograph, it is always what? It is always magnified. It is always magnified. And how many times you can magnify using the light microscope? You can magnify an object up to 1500 times, 1500x. So that is a very huge magnification, 1500 times power of your eye. So now the 1500 times power of your eye, suppose that you are given, and another thing, another very important thing is that when you give, when you are like magnifying something, you also lose the actual size of uh, an of a part of specimen. Suppose that this is an epidermal cell. These are the epidermal cells, and these are the guard cells. This is the total length that is given in the photomicrograph. Remember, this is a photomicrograph from X to Y. Okay. Now, what happens that when you magnified it, you have lost the actual size of a specimen. Why? Because the stomata, the size of stomata can never be equal to the size in an observable image. No, size of stomata cannot be, like, you cannot say that it would be this much larger. You getting what I'm saying?
Yeah.
So if it cannot be this much larger, it means that you need to know another thing, like what is the actual size.
Okay. So the
No, hold on, I need to tell you one more thing, then you can share your idea. Okay, the uh, the length between X and Y is the image length, like the length that is provided in the image. So it means it is not actual length, right?
yeah.
It is the length of the stomata provided within the photomicrograph. So it is the image length, not the actual length. So every picture that is provided has three different parameters. It has three different parameters. It has image length, actual length, and magnification. M. Actual size is A, and image length is I. Now we can stop here if you want to ask something.
No, I was just about to mention the magnification. This is basically magnification, right?
Hmm. So, what happens in the exam? They can ask you to find out both the things. They can ask you to find out the magnification of an object, of a given picture, or they may ask you about the actual size. Because the image length you can always measure. So for magnification, you would go M equals to I over A, and for actual size, you would go A equals to I over M. That is going to be your formulas. So what would you do here? You would go, first you would measure image length, measure I. Then you would be provided with the magnification, provided with magnification or actual size. Magnification or actual size. Remember, you would always be provided. You can never find the magnification by yourself or actual size by yourself. So you would always be provided with the magnification or actual size. You would just have to you would they would just hide one thing. Either they would hide magnification or they would hide actual size, but one of them has to be provided. So once they are provided, you would have to put them in the formulas, and then you can just simply calculate it. The most important thing about this particular question is the conversion of measurements. Remember, mostly actual size is in micrometer. Since it's a microscope, so remember that actual size mostly is in the micrometer. Okay, so what about the image length? Okay, when you would measure the image length, you must know that image length would be measured the ruler by the ruler, and most of the time the ruler has centimeters on it. The ruler has centimeter on it, not micrometers, not millimeters. So if you want to perform this, if you want to do this I over a, suppose I is y centimeter. So y centimeter over x micrometer. So this calculation would be wrong. What would you have to do? You would have to convert the image size into the micrometer. Never convert the value given in the micrometer into the millimeter or the centimeter. No, you have to go you have to go towards the micrometer always. These calculation would always be in the micrometers. At least we can say for CIEs that the calculations would always be in micrometers. So you never go from micrometer to millimeter or micrometer to centimeter, but you must now y centimeter. Y centimeter equals to y into 10. It would become y millimeter, not y millimeter. Suppose it becomes uh, a millimeter. A millimeter into 1000, it would become b micrometer, meaning the value is changing. So the value that you measured was y. Then you multiplied with 10, it turned into millimeters. And then you multiplied with 1000 and it turned into micrometers. So then you would perform the calculation. You would now do b. So then you can calculate the whole value. Got it?
Yeah.
Good. So this kind of question, um, they usually, very much usually appear in the exam, in the P4, sorry, P6. So let's try finding out a question or a P6 past paper, and let's do some practice for this particular um, paper, this particular concept. Mm. In this one, they have not provided the question. So let's try 62 summer 23. Hmm? This was basically a pet question of every paper before 2023. So you can imagine basically that since the students started guessing that this is one of the most confirmed question in every
No,
paper, they did
no.
not ask in 23. Hmm. Okay, so over here, yes, vascular bundle P to Q. P to Q is the length available, and this must be uh, stock of celery. Now, celery, you have been provided with the length P and Q. Now, what they are saying represent the width of celery stalk cross section. The actual width of the cross section is 27 millimeter. Now, you have been provided with the actual length. Since you have been provided with the actual length and it is in millimeter, then you would first have to find out the image length or length of PQ that you measured, and then you would be performing the calculation. So if you have a ruler, I would like you to measure the length of this. You got it?
Yeah, seven
Do you have a ruler?
centimeters. Seven.
Only 7 centimeters? Seriously?
Maybe it's because of the
Oh, it's
scale on the
centimeter.
computer.
Okay. So basically now it's going to be 70 millimeters.
Yeah.
So 70 millimeters. Now, since they are not giving you the actual length in micrometer, so you can simply put the values in the micrometer. So 70 divided by 27. How
Um,
much? What was the answer?
70 divided by 27, 2.59.
2.59 millimeters. It's going to be the length or the width of the total stalk. That is how you are supposed to be doing the question, or maybe some other questions like this would also could also be asked. So let's try some other one. It does not mean that this question would only appear in P6. They sometime give it to you in the P1 as well. So yes, it can be asked in the P1. Okay, so line AB is here. Okay, so length of line AB. Now you have to measure the magnification here. So what magnification you are going to tell? Length of the line AB in figure and actual width of the shell. So actual width is provided, I guess, 130 millimeter. You would measure the length of the line. So let's see.
Okay, so you ruler again.
Mm-hmm.
7.2 centimeters.
7.2 centimeter. So 7.2 centimeter means uh, 7.2 centimeter means it's going to be 72 millimeters. So 72 divided by the actual width. Uh, 72, 72 millimeter.
Yeah.
It's much less because they are providing the width of it. Okay, whatever. 72 divided by 130, just do it.
0.55.
0.55 multiplied with 1000.
55, 550.
72 divided by 130, 0.55. Now this is 0.55 mm. Okay, you need to tell the magnification, and magnification would always be told in micrometer. So 0.55 into 1000, it's going to be 550 micrometer, and in the end you would write 550x. Or maybe since it's a shell, you can stick to this magnification, because shell is much larger. It's not a microscopic object, right? So it's much larger than that, so you can stick to 0.55x. It also depends upon the specimen provided. Got it?
Yeah.
So that sums up your chapter of uh, cell, because the cell chapter was also here. We needed to finish this one. 0.62. So we were we're very close. Next chapter is about the movements of substances. Okay, so have you studied this chapter already?
Yeah, but I think I need to revise it.
How many chapters have you studied already?
Two. So, the other one is nutrition.
You told me you have learned about the classification as well.
Yeah, classification and then nutrition. I don't think I've done the movement. I don't think
Oh,
so.
okay, okay. Okay, so any idea about this one?
No, not really.
See, what happens that we always say the cells are the basic unit of life, right? We always say that it is the cells that perform the basic functions of life, right? Suppose there is a function of life called as respiration, in which we break the glucose into water and carbon dioxide. Do you know about this reaction?
Yes.
What do we call it?
The reaction.
This
So,
reaction, what's the
glucose,
name of this reaction?
um, I don't know the name.
Okay. The name of this reaction is called as respiration. And in this reaction, we make energy. You may be thinking that isn't respiration the breathing, right?
No, you told me that it's not breathing.
Well done, great. So, respiration is a process that is done inside the mitochondria. Yeah, I told you there when I was teaching you the mitochondria. And the now the carbon dioxide is produced in this reaction. The carbon dioxide is a waste. Similarly, you know that to perform this reaction, one thing is necessary, and that is the oxygen. So, now suppose this is a cell having this particular nucleus with this mitochondria, and the mitochondria, in the process, it is continuously making the carbon dioxide, and it is continuously requiring the oxygen. So the real question is how the substances how the substances are entering and exiting out of a cell. How is this happening? If the oxygen is entering, if the carbon dioxide is leaving, then why and how? What is making them entering and what is making them exiting? That's the real question that we'll be answering in this particular chapter. Got that?
Yeah.
All right. So, thank you very much. We'll meet.
Okay. Next week,
Allah.
Tuesday or Monday?